Uh, Good afternoon. I'm Sholotan Kazeem, here to discuss the comparative analysis of synthesized waltheria indica leaf and euphorbia maculata zinc nanoparticles. So we'll be looking at how, the green synthesis of zinc nanoparticle from these two plants actually helps in biological sciences. So these are the outline. We'll look at the introduction, aim and objective, materials, result analysis, and discussion, then probably the conclusion. So nanoparticles are actually being known to be materials that are of smaller sizes, usually measured in nanometers. So they range from 1 to about 100 nanometers. And zinc nanoparticles has actually been discussed severally, but there are limited information on the activity on many other type of microorganisms aside E. coli and more fewer on Staphylococcus aureus, but it is surprisingly that there are little information on their effect on fungi as well. So we'll be looking at the aim of this particular study. So the aim of this research, firstly, is to synthesize zinc nanoparticles and actually achieve this aim through the following objective: the crude extract using hot percolation method, then to determine the phytochemical constituents of both plants, also to synthesize and also characterize the zinc nanoparticles made from those materials using the Q250 scanning electron microscopy, 6405 Genway UV visible spectrophotometer, then the Fourier transmission infrared spectroscopy. We'll also compare the antimicrobial efficacy of the individual extract alongside that of the nanoparticle synthesized and some other conventional antibiotics. So it is also important to state that the antioxidant, anticoagulant, and addictive properties of those synthesized nanoparticles are also examined. So these are the materials and the methodology adopted for the process. So the plants of waltheria indica were collected and that of euphorbia maculata, the phytochemical processes, the zinc nanoparticle synthesis, instrumental characterization using the various equipment, then the anticoagulant activity of waltheria indica as well as that of euphorbia maculata zinc nanoparticles. So the antioxidant using the DPPH radical circumvention activity. So the next one here shows the result, the phytochemical screening. The result for waltheria indica leaves shows it contains tannin as well as that of euphorbia maculata. So in essence, they all contains taurine, saponin, glycoside, phenol, polysterol, terpenes, alkaloid, and protein, which is also evident from the FTIR conducted based on the bands and the transmittance of compounds present during the the FTIR analysis. Firstly, we look at the UV visible spectrophotometer characterization of the zinc nanoparticles formed, and it was observed based on literature. It is usually between 120 to about 380, though literature actually varies where we have the peak absorbance for zinc nanoparticle, but for this particular one, we got it at 380 nanometer with a peak absorbance of a 0.133, then which is actually for that of the waltheria indica zinc nanoparticle. And I think I actually omitted mentioning the type of material used in the synthesis of this particular material. It is zinc sulfate heptahydrate. So we actually mix it with the solution and the color changes based on the organogram of that of green synthesis. So this is for the euphorbia maculata. The peak absorbance actually differ from that of waltheria indica. That of waltheria indica is 0.133. Why this is at 0.153 at 380 nanometers. So the wavelength is actually specific for both two nanoparticle synthesized. The Fourier transmission infrared spectroscopy analysis. So it is evident enough that they both contain the amide group as well as the carboxylic stretch. But the only thing that actually differs here is the the transmittance level, the band available for both sides, which is also indicated in the material available here. So The oxygen-hydrogen stretch is also an indicative of what's it called? Hydroxyl group present. Likewise, we have the carboxylic, stretch. So these are actually present at this transmittance level. We have that of 3,265 per centimeter for 42.6%, which is the relative abundance, while that of the 329 per centimeter is at 75% abundance. So it is actually visible from the chart available here. So around this region, sorry, please, I don't know how to get the pointer. So, okay. It's actually available here, likewise here. So at this point, it is about 2,000 plus, which is also evident in the explanation actually provided there. And we have it to be around this region here. This is it, which represents the, what's called the CH2 group available in the compound. So moving away from the FTIR, what actually differs here has to do with the relative abundance of the matter, and what is actually responsible from it is a result of the nanoparticle synthesis that was actually formed. So we are to look at the next one. This is for that of waltheria indica crude extract, then in comparison to that of the zinc nanoparticle. So the first one has to do with the raw extract, the crude extract, while the other one here is that of the zinc nanoparticle. So the transmittance level definitely changes as a result of the zinc nanoparticles that have been formed in the solution. The next one here has to do with the same analysis conducted to determine the sizes of the synthesized nanoparticle. What actually surprises is that it was actually done to get the micrograph at 150 1 times 150 times 500 and times 1000, but I actually have in this particular structure the micrograph of that of times 200 micrometer. So in the course of what's it called, using ImageJ and Guidon, a software analysis through which the sizes of the various or the individual nanoparticle is actually being obtained. So we have it here using ImageJ, and from there we're able to determine the various sizes, the diameter sizes of these various nanoparticles. So the ranges of this nanoparticle is actually captured here to be within a value of 3.766 nanometer to a value of, 6.808 nanometer. That is the ranges of the nanoparticle synthesized from the, method. So for that of the waltheria indica, we also have it to be from 11.87 here to a maximum value of 87.2 nanometer. The good thing about it is such that the nanoparticle synthesized of very small sizes that it could help in the delivery of drug substances into the cells. And interestingly, we have it here to be the antimicrobial susceptibility test, but I think I will have to skip this. Let's go to some, other part of it, the antioxidant activity. Then I'll move back to, the antimicrobial analysis. It is observed that at different concentration of the various extract, the crude extract and the nanoparticle that are being synthesized, it is being observed that at a very high concentration of 0.1 milligram of the solution, as in of the substance, into 0.2 mil, the euphorbia maculata extract actually had a kind of absorbance of a 0.45, and the interpretation of the DPPH activity here is at 34.54, and it actually decreases down because it shows that there is no more scavenging activity at 0.4 concentration and a 0.6 concentration for the crude extract. But going straight to that of the zinc nanoparticle, it is observed that at 0.2 concentration, we have it to be 23.3, where we have no scavenging activity at all for that of the zinc nanoparticle. It becomes a increased to a kind of 18.72, which show that this particular zinc nanoparticle has actually improved the antioxidant activity of the initial crude extract. Likewise, we also have it here to be 21.19. The funny thing about this is such that at a particular time, we have a decrease in the value of the scavenging activity and also a sharp rise again from, 18.72 to 21.19. So it actually shows that the, concentration decreases. There's possibility that we continue to have a very higher activity of, antioxidant effect of this euphorbia maculata. This is a kind of weed that is commonly available on a rocky part or a kind of a pavement. So then for the waltheria indica, the crude extract was observed to have a better antioxidant effect than that of the ascorbic acid, which was used as a kind of the control, because we tend to observe that this waltheria indica extract had a better type of antimicrobial, antioxidant activity than ascorbic acid of 68 point something thereabout. Okay, not saying clearly, 68.67, I guess. Then we also observe here that the waltheria indica extract decreases just the same way the ascorbic acid scavenging activity also decreases. But in the case of the waltheria indica zinc nanoparticle, though it actually declined from 72.71 to 19.45. But similar trend observed in that of euphorbia maculata is also seen in the case of zinc nanoparticle, because we observe it actually have a sharp decline from 19.45 to 4.64, and also a kind of rise again to 9.0. So just to eliminate the guess that probably the zinc sulfate solution could be responsible for the activity. So it's also used as a form of control to determine the scavenging activity of the zinc sulfate heptahydrate alone to see if what probably the reaction was what was bringing out. And it is evident enough that here we have 0. Scavenging activity here, a further decrease, showing that the zinc sulfate solution had no business in that. It's only as a result of the nanoparticle that has been synthesized from the combination of the crude extract and that of the zinc sulfate solution forming the nanoparticle were responsible for the scavenging activity available here. So we also try to check the antimicrobial activity of the synthesized nanoparticle against this selected organisms: Aspergillus niger, Aspergillus flavus, Escherichia coli, Klebsiella pneumoniae, Pseudomonas aeruginosa, Proteus mirabilis, and Staph aureus. And surprisingly, we tend to see, though not kind of pronounced, antibacterial activity, but they are of, I think, prospect if further, reconcentrated to see at a higher concentration. Oh, sorry, please, let me just quickly round up. So at this point, it showed that there are some antimicrobial activity of the synthesized nanoparticle, even when compared to fluconazole, which are normal or standard drugs for fungi, while we also have ciprofloxacin and gentamicin control. And there are some cases whereby we have the zinc nanoparticles having a better efficacy than what gentamicin is actually providing. So the next one, again, we actually check, okay, this is a kind of pictorial information, graphical illustration of the DPPH. This is the anticoagulant activity. So what we did here was just to check between the EDTA and these nanoparticles, which one had more anticoagulant effect. And we observed that the mixture of blood sample with the individual zinc nanoparticles had better effect than that of EDTA, because EDTA was able to last for 180 minutes, while the waltheria extract, the zinc nanoparticle of waltheria and that of euphorbia were able to stand for about 480 minutes upon observation in the laboratory. So the next one, this is a kind of graphical illustration. So what we also need to check is, again, the addictive properties of the synthesized nanoparticle that is in Nigeria, where I actually come from. Many times you paint your building and over time you observe some molds growing around it. So how do we tend to prevent the growth of molds around kind of a building? This is a kind of direct application. So we actually obtain some paints and we mix them with what's it called, different organisms, such as Klebsiella pneumoniae. We also mix that with Staph aureus and Aspergillus niger. So those were the organism we actually carried out the studies on. So it was observed that the nanoparticles had a better addictive effect against Aspergillus niger by reducing the growth of the organism to a very scanty population on the plate. This is a kind of plate that contains the mixture of the nanoparticle, the microorganism itself, and paint. So every paint, we have a solution, a kind of culture of the organism, which is being mixed with the paint, coupled with 1 ml of what's it called, the zinc nanoparticle solution, and it's been allowed to stand for some time. Then that particular, what's it called, 0.1 ml of the mixture of paint, nanoparticle, and the organism is actually being plated to see if there will be enormous growth on the plate after incubation. So for the case of waltheria indica, we observe there is no growth at all. So it showed that waltheria indica has a very good ability to inhibit the growth of Aspergillus niger, thereby preventing a kind of spoilage, or what's it called, the facing of a painted wall. Why that of euphorbia maculata has a kind of reduced, but not as perfect as that of waltheria indica. So for the next one, we have that of Klebsiella pneumoniae. Though considering the plates count, it was observed that that of waltheria indica also had a kind of limited growth compared to what is obtainable. The euphorbia maculata zinc nanoparticle is more effective against the gram-negative as compared to waltheria indica zinc nanoparticle. So for the case of Staph aureus, it is evident enough that the euphorbia maculata could not prevent the proliferation of the Staph aureus, while waltheria indica was able to reduce to a very limited extent. So it's a kind of direct application of the zinc nanoparticle. So in conclusion, the synthesized zinc nanoparticle showed varying antimicrobial activity against the test isolate. They have high radical scavenging activity, high coagulant effect, and as a result, they could be a prospect of broad spectrum antibiotics, anticoagulants, antioxidant addictive effects, instead of we concentrating, because the trend in microbiology has always been the deployment of these nanoparticles towards antimicrobial effects. So checking out, do we have some other attributes, multifunctional, like presented by the earlier professor, showing some other functions that can be performed by synthesized nanoparticle. Though the shortfall of this study is that we were unable to carry out the toxicological effect of the plant as well as the synthesized nanoparticle, which is an area that needs to be worked upon in the next work. So this happens to be a master's research conducted in 2018. Thank you.